In this lecture series, we are going to be taking a look at glycogenolysis and glycolysis. Glycogenolysis is the breakdown of glycogen within cells. We're going to look specifically at how skeletal muscle cells do this, but a lot of this is applicable to other cells that store glycogen as well. Glycolysis is the breakdown of glucose, largely to form ATP and some other molecules that we'll discuss throughout the lectures. The reason that we're discussing these two concepts together is because it logically makes a lot of sense to do so. The reason for that is that glycogen is the storage form of glucose inside of muscle cells and other cells. Glycogen is a polysaccharide. We have discussed this to some degree in other lectures. We've talked about how glucose can be transported through the bloodstream and uptaken by muscle cells and other cells and then stored as glycogen, right? So this right here, this glycogen here is a polysaccharide. It's a whole bunch of glucose monomers all bound together. If you were to look inside of a skeletal muscle cell after a high carbohydrate meal, you would see lots of what are called glycogen granules. They look like little pebbles inside of the muscle cell, and they're all glycogen. And then if you were to look at those same skeletal muscle cells, let's say a couple hours later, after really high intensity exercise had taken place, there would be a lot less or even no glycogen granules left. So that must mean that during exercise, higher intensity exercise, the muscle cell consumes all of this glycogen. So what's actually happening there? What's happening is during exercise, glycogen is being broken down. That process is called glycogenolysis. And the reason it's being broken down is because when you break glycogen down, you have to think about what glycogen is made up of. It's made up of a whole bunch of glucose monosaccharides. So if you break glycogen down, you free up a whole bunch of these glucose monosaccharides. And these glucose monosaccharides can then be used in glycolysis, which serves the purpose of making ATP, as well as other molecules that the muscle cell would need during this instance of exercise. So the relationship between glycogenolysis and glycolysis is fairly obvious. If a muscle cell wants to run glycolysis, right, the breakdown of glucose to make ATP and other molecules, then it's also going to need some glycogenolysis to be occurring. It's going to need some of the glycogen within it to be being broken down so that glucose monosaccharides can become available so that it can run glycolysis. So in part one of this series, we're going to take a look specifically at glycogenolysis. How is glycogenolysis initiated? How is it initiated, from what circumstances, and why? And we're going to focus on skeletal muscle cells, and we're going to use exercise as a means to explain it. So the reason I'm using exercise as a means to explain this is for somewhat of a dramatic effect, and I think people in some ways can relate to it a little bit better. And even if you don't exercise much, I mean, just imagine doing physical activity, perhaps. During instances of where muscle is contracting more forcefully and more frequently, glycogenolysis happens at a really rapid rate. So let's answer why, first of all, and then how. So in this instance, we are looking at a race. Let's pretend that this is a sprint. We're going to use a sprint as an example. Okay. During a sprint, within skeletal muscle cells that are involved in the sprint, the rate of glycogenolysis is going to be really great. It's going to be really fast. Why? Well, as I've already said, what happens during a sprint is the muscle cells begin to contract really forcefully and really frequently. And in order to do that, they begin to consume lots of ATP. Okay? Because muscle cells, in order to be able to contract, they have to consume ATP. The more they contract, the more forcefully they contract, the more ATP they'll consume. So I think it's somewhat logical to think or realize that during a sprint, muscle cells consume a lot of ATP. Now, every cell in the human body plays a pretty delicate balance, or has a pretty delicate balance that it has to play, which is it really, all the cells have to make as much ATP as they're consuming. Otherwise, they have the potential to die, the cells themselves. So I tend to say ATP consumption is the same thing as ATP demand. Okay? However much ATP the cell is consuming, right, that's the amount of ATP that it's demanding in that circumstance. And that ATP demand needs to be met. Another way of saying that is, the muscle cells need to make just as much ATP as they're consuming. And in the instance of a sprint, that's a lot. So one thing that's really useful for the muscle cell to run when the ATP demand is very, very high, which I will explain through the series of these lectures, is glycolysis, right? The breakdown of glucose, particularly a type of glycolysis, which we'll discuss. But that is very helpful for meeting high ATP demands. So during this instance of a sprint, the ATP demand within this person's skeletal muscle cells is very high. And that means those muscle cells need to make a lot of ATP. And the best way to do that, there's a couple good ways to do this, but one of the best ways to do that is to run glycolysis. Glycolysis, again, particular types of glycolysis are very good at making lots of ATP really quickly to meet high ATP demands. Well, how are these skeletal muscle cells going to start running all this glycolysis to meet these high ATP demands? Those muscle cells are going to have to break down glycogen. They're going to have to perform glycogenolysis because that will free up the glucose monomers to be consumed in glycolysis to make that ATP to help meet the ATP demand. So hopefully on an introductory level, this helps you answer to some degree the why portion of this. Why during a sprint would muscle cells need to rapidly run glycogenolysis, the breakdown of glycogen? Well, to free up glucose so that it can be consumed in glycolysis to meet high ATP demands, which the muscle cells are experiencing during the sprint. But now we need to do the harder part, which is explaining how this happens. How inside of the muscle cells does the rate of glycogenolysis increase so much? So there is this enzyme that I've discussed with you a little bit in the past, not much, but glycogen phosphorylase. And what glycogen phosphorylase does is it breaks individual glucose monosaccharides off of the glycogen chain. Specifically, what it does is it strips one molecule of glucose off of glycogen adds a phosphate to it and turns it back into glucose 1-phosphate. And then glucose 1-phosphate can be turned back into glucose 6-phosphate. Okay, so glycogen phosphorylase is the enzyme that we need to activate if we want to get glycogen to be broken down at a faster rate, so that we can then run that glucose that we're breaking off of glycogen through glycolysis at a faster rate. So the question for now is, how is glycogen phosphorylase regulated? Because if we can increase the activity of glycogen phosphorylase, what are we going to increase? We're going to increase the breakdown of glycogen back to glucose, specifically glucose 6-phosphate, right? We're not turning glucose back. We're not turning glucose from glycogen actually back into glucose, because then the glucose could actually leave the skeletal muscle cell, and the skeletal muscle cells don't have the capacity to do that. What we're doing is we're breaking glucose off of glycogen and turning it back into glucose 6-phosphate. So there are two pathways primarily that increase the activity of glycogen phosphorylase, which then allows for more glycogen to be broken down. Now, what those two pathways do is they increase the activity of something called protein kinase A, or pKa, and the activity of what's called calcium calmodulin dependent kinase. A simpler way of saying this is that if you can increase the activity of either pKa or calcium calmodulin dependent kinase, then glycogen phosphorylase will become more active and more glycogen will be broken down, allowing for glycolysis to run at a faster rate. So what we need to do is take a look at the pathways that increase the activity of protein kinase A and calcium calmodulin dependent kinase. We're going to start off by taking a look at the pathway that increases the activity of protein kinase A, and then how protein kinase A increases the activity of glycogen phosphorylase. It should be no surprise that under the instance of a sprint, these two pathways are going to be heavily upregulated. So let's now take a look at that. Okay, so just with the anticipation of exercise, what's going to begin to happen is the adrenal glands are going to begin to release epinephrine into the bloodstream. Okay, so epinephrine levels are going to begin to rise just with the anticipation of exercise. And by the time that you're exercising, actually physically exercising, and in my example, half a second into that sprint, epinephrine levels within the bloodstream are going to be pretty dramatically elevated from what they are at rest. All right, so what is one of the things that epinephrine does? One of the things that epinephrine does is it binds to integral proteins receptors located on your skeletal muscle cells. So here is a skeletal muscle cell, plasma membrane of it, and what I'm depicting here, I'm showing you, this is not actually what the image is showing you, but I'm showing you this. I'm showing epinephrine, which is elevated in the bloodstream and thus the interstitial fluid. During exercise, I'm showing you that epinephrine binding to an epinephrine receptor on a skeletal muscle cell called an adrenergic, that stands for adrenaline, adrenergic receptor located on the skeletal muscle cell within the plasma membrane of the skeletal muscle cell. Okay, so what does epinephrine do when it binds to this adrenergic receptor? What it does is it activates a G protein pathway. Let's talk about that in some more detail, excuse me. There are a variety of different G protein pathways. The one that I'm going to focus on right now is the cyclic AMP G protein pathway. So with the cyclic AMP G protein pathway, first of all, you need the ligand, in this case epinephrine, to bind to the receptor. The receptor is an integral protein. And then attached to the receptor on the intracellular side are more integral proteins. Okay? What are these integral proteins? There's an alpha subunit, a beta subunit, and a gamma subunit. All right. So in the case of the cyclic AMP pathway, what happens is, first of all, when epinephrine, or whatever ligand this is, binds to the receptor, okay, the alpha subunit dissociates away from the beta and the gamma subunit, by swapping out GDP for GTP. Okay, so the alpha subunit swaps out GDP for GTP, and then it dissociates away from the beta and the gamma subunit. And then what the alpha subunit does is it travels along the intracellular side of the plasma membrane, and it activates an enzyme that's actually another integral protein. This is an integral protein that actually serves as an enzyme called adenylate cyclase. And what adenylate cyclase begins to do is it begins to turn ATP into cyclic AMP. Okay, so what we do, what actually is involved here is you strip 2 phosphates off of ATP, which makes AMP, adenosine monophosphate. If you remove obviously 2 phosphates off of adenosine triphosphate, you're left with adenosine monophosphate. That's where the AMP comes from. And cyclic or cyclic, this actually gets turned into the shape of a ring. The remaining phosphate actually gets bound to the sugar within adenosine monophosphate, and thus its actual molecular shape is more cyclical, and that's where the term comes from. Okay, so adenylate cyclase's job is to turn ATP into cyclic AMP, and that's exactly what it does when it's activated by the alpha subunit. Okay, so what does cyclic AMP do? Cyclic AMP is called the second messenger. What cyclic AMP does is it goes over and it activates pKa, which is protein kinase A. I just showed you that pKa influences glycogen phosphorylase. Okay, so pKa is activated by this cyclic AMP pathway. So let's briefly recap and realize what's happening inside of this skeletal muscle cell right now during exercise. So just in case you wanted any more clarity on cyclic AMP, here is ATP, adenosine triphosphate, right? Adenosine and 3 phosphate groups. Okay. Over here, AC stands for adenylate cyclase. What adenylate cyclase does is it takes ATP and first of all strips 2 of the phosphate groups off. Well, if 2 phosphate groups are stripped off of ATP, you now have what's called AMP, adenosine monophosphate, right? You've got adenosine plus 1 phosphate group, adenosine monophosphate. Okay? But what also happens in the reaction is the remaining phosphate group is covalently bound now to the sugar, right, ribose. And this creates a sort of cyclic structure, so the molecule is now called cyclic AMP. And it serves as one of the most important second messengers in the human body. Here you can see epinephrine binding to the receptor, right? This is called an adrenergic receptor. It's an integral protein that serves as a receptor. Okay, when epinephrine binds to this adrenergic receptor and activates it, right? This adrenergic receptor is a cyclic AMP G protein pathway. Okay, so here's what happens, right? You know a lot of the process here. Epinephrine binds to the adrenergic receptor, and when the adrenergic receptor is activated, the alpha subunit will swap out GDP for GTP, travel along the plasma membrane, the intracellular side, and activate adenylate cyclase, which more realistically should be up here. And then what adenylate cyclase does, adenylate cyclase begins to convert ATP into cyclic AMP. And then what does cyclic AMP do? Cyclic AMP activates protein kinase A, or pKa. Okay. So this should tell you something. When epinephrine levels rise inside of the bloodstream during exercise, protein kinase A levels within our skeletal muscle cells will increase. And just for clarity, this is showing cyclic AMP actually putting pKa into its active state over here. So you would summarize this in saying that epinephrine binds to skeletal muscle cells, and that binding of epinephrine to skeletal muscle cells, specifically to adrenergic receptors on the skeletal muscle cells, causes an intracellular increase in the activity of protein kinase A, right? And that's thanks to the cyclic AMP pathway that the G protein adrenergic receptor is. So now we need to figure out what pKa, protein kinase A, does. Look at the K, that stands for kinase, as I've said many times. So pKa is a kinase. That means that pKa is going to go around and start phosphorylating things. So what does pKa, in fact, phosphorylate in order to influence glycogen phosphorylase? All right, so here is epinephrine binding to the receptor, adrenergic receptor on the skeletal muscle cell, which is ultimately translating into adenylate cyclase, converting ATP into cyclic AMP, which is activating pKa, okay? So epinephrine is causing a rise in pKa, protein kinase A, and then what does protein kinase A do? Well, it phosphorylates what is known as phosphorylase kinase. And it makes phosphorylase kinase active by phosphorylating it. So pKa phosphorylates phosphorylase kinase, making phosphorylase kinase active. And then what phosphorylase kinase does is phosphorylase kinase phosphorylates glycogen phosphorylase, and that makes glycogen phosphorylase active. And you know what glycogen phosphorylase does. Glycogen phosphorylase begins breaking glycogen down back into glucose, specifically glucose 6-phosphate at the end. Okay, so this is one of the ways that the breakdown of glycogen increases during exercise. It's thanks to the increase in epinephrine within the bloodstream. Specifically, epinephrine binding to adrenergic receptors on skeletal muscle cells, which are cyclic AMP G protein pathways that increase the activity of pKa inside of those skeletal muscle cells. And if you follow that downstream, you can see that a rise in pKa activity inside of skeletal muscle cells, thanks to epinephrine, results in an increase in the activity of glycogen phosphorylase, which begins to break glycogen down into glucose, of course, at a faster rate when glycogen phosphorylase is more active. So as far as glycogenolysis goes, the breakdown of glycogen, we have taken a look at the first pathway that would increase its rate, right? We would need to increase the activity of pKa, which is accomplished largely by epinephrine, which then increases the activity of glycogen phosphorylase, which causes more glycogen to be broken down. We, however, have not looked yet at the other pathway that increases the activity of calcium calmodulin dependent kinase, which can also then increase the activity of glycogen phosphorylase and cause glycogen to be broken down as well. So that's what we're going to do now. So I've been basing this whole conversation around the idea of a sprint. Okay, so again, during a sprint, skeletal muscle cells contract really forcefully and frequently. And in order for skeletal muscle cells to be able to contract, yes, they have to consume ATP, but also they need calcium levels to elevate inside. And maybe you can imagine, the more forceful and frequent a contraction is, then the more calcium there's going to be inside of the muscle cell. And what will happen, this is unrelated to how calcium allows for the muscle cell to contract, but when calcium levels elevate, some of the calcium will bind with what's known as calmodulin. And when calcium binds with calmodulin, it forms what's called a calcium calmodulin complex. And then calcium calmodulin, the complex, will bind to and activate what are called calcium calmodulin dependent kinases. So calcium calmodulin dependent kinases, there's a variety of different kinases. Rather than going through all the different ones, we'll just say calcium calmodulin dependent kinase. We'll make it singular. The activity of calcium calmodulin dependent kinase will be significantly upregulated. right? When the muscle cell starts to contract during the sprint, calcium levels will elevate inside of the muscle cell. Then calcium will bind with calmodulin. Some calcium will bind with some calmodulin, form these calcium-calmodulin complexes, which will then activate and increase the activity of calcium-calmodulin-dependent kinases. If you're wondering where the name comes from, just think about it. Kinase, again, implies an enzyme that phosphorylates things. Well, this kinase enzyme is calcium-calmodulin dependent, meaning it needs calcium-calmodulin to bind to it in order to become active. Calcium-calmodulin dependent kinase. So now it's very active. And what might it be able to do?
So what is calcium-calmodulin dependent kinase? It's a kinase. So it phosphorylates things. And specifically, it looks like inside of skeletal muscle cells, calcium-calmodulin dependent kinase can phosphorylate and activate glycogen phosphorylase, which you know begins the breakdown of glycogen, right? It breaks glycogen back down into glucose, specifically glucose 6-phosphate. So calcium calmodulin increases the activity of glycogen phosphorylates, which it's going to break more glycogen down into glucose. So what is this telling us? This tells us that whenever we ask skeletal muscle cells to contract, and this will happen proportionately to, how frequently and forcefully we ask those muscle cells to contract, what's going to happen is when those skeletal muscle cells contract, calcium will increase inside. Calcium will bind with calmodulin. And calcium calmodulin will then activate calcium calmodulin dependent kinase, which will phosphorylate the enzyme glycogen phosphorylase and make it active so that more glycogen is broken down into glucose. So collectively, this implies that when skeletal muscle cells are asked to contract, those skeletal muscle cells, thanks to an increase in calcium on the inside, are going to begin to break glycogen down at a faster rate. So this is an actual completely intracellular pathway, we could say intrinsic pathway, that exists inside of skeletal muscle cells to increase the breakdown of glycogen to glucose during exercise or when those skeletal muscle cells are being asked to contract more. So let's take a step backwards and talk about what all of this means that we've discussed so far for our sprint. So what I said at the very beginning was that half a second into a sprint, the active skeletal muscle cells that are involved in that sprint, within the quadricep, hamstring, gluteal muscles, etc. Those active skeletal muscle cells, half a second into the sprint, are already breaking lots of glycogen down to glucose. And the reason that those active skeletal muscle cells within the sprint are already breaking lots of glycogen back down into glucose is because their glycogen phosphorylase activity has dramatically increased already half a second into that sprint. And that's what's breaking the glycogen down to glucose. But what's causing the glycogen phosphorylase activity to increase? Now, in more basic courses, what I say to people is that epinephrine rising in the bloodstream is involved in activating glycogen phosphorylase, and I say that calcium increasing inside of the skeletal muscle cells is also involved in activating glycogen phosphorylase, and thus that is why the skeletal muscle cells are breaking more glycogen down into glucose. What I bypass in those courses is explaining how epinephrine actually causes the activity of glycogen phosphorylase to increase, how epinephrine rising in the bloodstream does that, and how calcium rising inside of the skeletal muscle cells during the sprint actually activates or increases the activity of glycogen phosphorylase. And the answer, I hope you know now, is that epinephrine binds to adrenergic receptors on the skeletal muscle cells, and that adrenergic receptor is a cyclic AMP pathway, which increases the activity of protein kinase A, and you follow that downstream, and you realize that protein kinase A activates or phosphorylates glycogen phosphorylase, which makes it active. And hopefully you also know that the reason that an increase in calcium within the skeletal muscle cell causes an increase in the activity of glycogen phosphorylase is because calcium binds with calmodulin. Calcium calmodulin activates calcium calmodulin dependent kinase, which then phosphorylates and activates glycogen phosphorylase. So what this has been is a much more detailed explanation as to how epinephrine rising in the bloodstream during exercise increases the breakdown of glycogen to glucose by activating glycogen phosphorylase, and a more detailed explanation as to how calcium increasing in the skeletal muscle cells during exercise also increases the activity of glycogen phosphorylase and breaks more glycogen down to glucose.
I'm going to finish by summarizing and setting us up for the next part of the lecture series. So the overarching point here is these muscle cells, or any muscle cells in anybody that are involved in a sprint, have very high ATP demands, right? They're consuming lots of ATP, so they need to make a lot of ATP. One of the best ways that they can make a lot of ATP to meet these ATP demands is to run glycolysis. The muscle cells want to run glycolysis. If they want to run glycolysis, then they need glucose. Where can they get that glucose from? Glycogen that's stored within them. But they need to break that glycogen down to free up the glucose monomers. And that's going to require the activation of glycogen phosphorylase to break down the glycogen. And the pathways that break down glycogen through the activation of glycogen phosphorylase are largely driven by epinephrine increasing in the bloodstream and calcium increasing inside of the muscle fibers. Epinephrine increases pKa levels, calcium increases calcium calmodulin dependent kinase levels, which then both have the ability to increase glycogen phosphorylase levels, which causes glycogen to be broken down. And this is going to happen really rapidly, the rate at which glycogen is broken down. And that's a proportional thing, because there is so much epinephrine in the bloodstream during a sprint and so much calcium within the muscle fiber during a sprint that there's going to be very high levels of pKa and calcium calmodulin dependent kinase within the muscle cells, which is going to activate lots of glycogen phosphorylase to a great degree, which is going to cause lots of glycogen to be broken down to glucose, which then, leading into the next lecture, is going to allow for glycolysis to happen at a really rapid pace. The muscle cells want to run glycolysis at a really rapid pace, and now, because they increase the rate of glycogenolysis so much, they've got a lot of glucose available to them so that they can run glycolysis at a rapid rate to make a lot of ATP to try to meet this ATP demand. So in the next lecture, we'll take a look at glycolysis. And specifically, we'll look at how glycolysis is run inside of muscle fibers during the instance of a sprint. So that's what's coming next.